All right, so today is Lesson 8, Alterations in Musculoskeletal Functioning. Good news is this is going to be a slightly shorter slideshow, and we should be out a little bit early, and everybody can be very happy about that. By the way, the quiz will be open today after class, again, open for 1 week on the last 2 weeks' worth of lessons. All right, so musculoskeletal system, this is going to be very familiar to most of you. Components are bones, joints, cartilage, ligaments, tendons, and muscles. The function is essentially support, protection of your internal organs, movement, mineral homeostasis. We don't often think of that, but your skeletal system is responsible for your mineral homeostasis, blood cell production, that's where all your blood cells are being produced, and your triglyceride storage. So when we think of the musculoskeletal system, we're essentially thinking bones, joints, muscles, and nerves. Any one of those, and you do not have movement. Two types of skeletal systems. There's the axial skeletal system and the appendicular skeletal system. The axial skeletal system is actually just simply there to protect your vulnerable internal organs. This is your skull, protecting your brain, your vertebral column, your thoracic cage that's protecting your heart and your lungs and your liver, etc. The appendicular skeleton is what we typically think of when we say skeletal system, and that is movement. So that includes your upper and lower, your upper girdle, your pelvic girdle, your lower and upper extremities. These allow you to move around. You do want to know the difference between these two, axial versus appendicular skeleton. So when we think about long bone is things like our leg bones and our arm bones. There's a very specific structure to the long bone. Essentially on the inside is that kind of spongy bone, the cancellous bone. On the outside is the cortex. And then we have kind of different regions, you want to call them geographic regions of the bone that we tend to refer to, particularly when we're reading x-rays or referring to things like fractures. So you'll have your proximal epiphysis. The epiphysis is kind of where the head joins to the long part of the bone. The diaphysis, which is your shaft. And then your distal epiphysis is where it joins to another joint. Again, proximal being near, proximity, distal being distance, further away from your core. So anytime you hear the words proximal and distal, proximal means close to your core, distal means farther away from your core. Muscles essentially connect bones via tendons and contract to provide the force that we need for movement. So muscles are going to connect a static bone to a bone that moves. So you can't have two static connections. You have to be able to contract and shorten in order to provide movement. As your arm bends, you've got a moving bone that's connected to a static bone that's being pulled by the muscle. Skeletal muscles are going to exist in antagonistic pairs. So pull up and a pair that allows you to extend back out with the opposite movement. So that's where we start getting into the things like flexion and extension that you're going to be assessing when you assess your patients, adduction versus abduction, etc. Always tendons connecting muscles to bones. Ligaments are not connecting muscles to bones. Tendons are. Ligaments are generally going to be bone to bone. So there are three main types of muscular tissue throughout the body. We have our skeletal muscle tissue, we have our smooth muscle tissue, and we have our cardiac muscle tissue. Skeletal muscle tissue is under our voluntary control. Smooth muscle is not. And really neither is cardiac unless you are somebody who meditates substantially and can slow your heart rate. So skeletal muscle is attached to the skeleton and it's responsible for all those voluntary movements. Smooth muscle are going to be things like the smooth muscle around your bronchioles, things that cause bronchospasm. The muscles that you cannot control, involuntary movement, breathing of the lungs, etc. These are non-striated and they have a different kind of shape to the muscle fibers versus what we think of the typical striated look that is associated with skeletal muscle. And then cardiac muscle we went into last week, 2 weeks ago. So when we think of how skeletal muscles are organized, it's pretty straightforward. It's bundles within bundles. They are tightly packed little muscular bundles that are surrounded by little kind of connective tissue sheaths. Think of this as almost like an electrical cable in a way, where you have a cable, plastic thing outside, and a bunch of little wires running down through the middle of it, and that's essentially what a muscle bundle is. So we have our big bundles, the fascicles, that are surrounded by the paramecium. And then inside of each of those, we have little muscle fibers that come together. These are called myofibrils, and those are the items that are specifically responsible for contraction. Myofibrils are going to be further divided into little repeating strands called the sarcomeres, and these are almost like bungee-type systems that hook together, and they're the units that actually do the sliding and the contracting over each other. And you'll see that if you look at the picture of the sarcomere, there's actually almost these little coiled ends, and there's kind of prongs sitting outside of it, and they hook into each other, and they actually do this almost paddle-like effect in order to draw those units together as they use energy. It's really quite fascinating if you look at it. So let's take a good look into a muscle fiber. So we have our myofibril, which is the big kind of red bands. It gives this kind of striated effect. Chondria that are there to help provide energy and ATP, which is necessary for this whole process. So you can see that it's kind of a slightly different effect when we look at these muscle cells than what we think of as a typical cell. Sarcomeres, again, that unit that does the actual contraction, is basically consists of thick and thin filaments, myosin versus actin. And you may have heard these terms in the past, myosin versus actin. What happens is that these filaments are moving in opposite directions over each other, creating a sliding type of effect. So they're sliding into each other as the muscle itself is contracting. And then there's these kind of like little Z lines that help to anchor everything and to hold it in place. Multiple sarcomeres, when they're layered on top of each other, is what gives us that characteristic striated look of muscle fiber. So the ends of the sarcomere tend to be a little bit lighter because only actin is present in that band. We don't have any myosin in that band, so it gives us a lighter appearance. And that's why muscle tends to look light, dark, light, dark, light, dark when we look at it. This is a big picture of a sarcomere. And essentially what's happening is these little actin filaments reaching forward with paddle-like kind of appendages that are hooking into the myosin and pulling and contracting it together. And that causes the actual shortening of the muscle. So muscle contraction has 4 steps. We depolarize our cell and we release calcium. You need calcium. If you do not have calcium, you cannot contract your muscles. That is why we start getting kind of hypotonic when we don't have enough calcium in our body. So if you don't have calcium, you're not going to be able to go through this process. Then what happens is we get a actin-myosin cross-bridge type formation. They start kind of making these little cross-bridge connections, and then they grab each other and they paddle in the opposite direction to kind of pull together, and the sarcomere itself shortens, which is essentially the contraction of the muscle fiber. All right, so here's just a picture of depolarization and calcium ion release within your muscle fibers. An action potential from a motor neuron is coming in, and it's triggering the release of acetylcholine, one of our major neurotransmitters, into the end plate. Acetylcholine is triggering depolarization within that sarcolemma, which then kind of follows a wave-like pattern down. This then causes that kind of shortening that we were just discussing. So that's your initial depolarization. This is a picture of your cross-bridge formation, where what happens with depolarization is that there's little myosin heads, and they're actually uncovered, and this is troponin. So you may have, obviously we're fairly familiar with troponin, but the calcium ion binds to the troponin, which exposes these little myosin heads and actually triggers the ability to create these cross-bridge formations so that you're pulling along that. And we need ATP for this action. So without ATP, without sufficient energy production, we can't create these kind of cross-bridge formations. And then we have this little kind of sliding mechanism that goes on. So this picture over here shows you almost this paddle-like effect where with ATP, the paddle is actually picking up, it's moving forward, and it's pulling it down, and that's causing the shortening of the muscle fiber. And that is showing you what it looks like once it's relaxed versus once it's contracted. You can see that the two ends actually come and they slide together so that it's shortened there. So that's the shortening of the sarcomeres, contraction of your muscle. So a summary of our muscle contraction is that an action potential in a motor neuron is going to trigger release of calcium ions. The calcium ions are going to bind to troponin, which is on actin, and that is going to expose these kind of myosin heads. And then the filaments between actin and myosin are going to form those cross bridges. ATP is going to come along. They're going to actually paddle each other forward, and we're going to get movement of the muscle and shortening. So that's the sliding mechanism that causes the muscle contraction. So 3 types of skeletal muscle fibers. I didn't say, not 3, excuse me, 2. 2 different types basically that we use for movement. We have our slow twitch muscles and we have our fast twitch muscles. Slow twitch is more for your endurance. They contract much more slowly and they don't fatigue as easily. Fast twitch is more used for rapid movement and muscular strength, but they do fatigue far more easily. So athletes actually can have a higher proportion of fast twitch muscles. People who are very, very good actually have a higher amount of this fast twitch muscle than those of us who are not perhaps as athletically inclined and who may have more of the slow twitch muscle. So they do actually have a physical advantage over other people.